moving on to section 5 already. We're going to blow through the last 2 sections, I feel like, because so much of the emphasis of genetics in high school is put on just kind of those basics that we talk about in sections 1 and 2. But if you guys recall, the last time we talked about DNA replication, okay? Something that happens within the cells in cell division. Remember? Mitosis and meiosis, don't forget those 2 things. So we talked about how DNA is replicated. Not that it, I guess I'm kind of, let me back up. It doesn't happen in mitosis and meiosis. It happens in interphase of both of those, okay? So remember that. But that's where we went with last time. Now, let's talk a little bit more about why, I think I mentioned, or actually it wasn't me, but I showed you guys a video about what happens if something is wrong in your gene code. If you have just one mistake in that gene code, then the proteins that it makes all of a sudden are very, very different. Well, I shouldn't say they're slightly different, but causes huge, huge effects. So basically the shape of the protein may change ever so slightly that you and I would never even notice in looks-wise, but it could change something so drastically that you have a disease such as cystic fibrosis or Tay-Sachs disease. So let's talk about the protein making part, okay? That's what we're going to get in today. So we're going to talk about how we get DNA to basically make proteins, and that's called protein synthesis, okay? A very, very important part of genetics, because obviously this is what codes for the specific things that we need in our bodies and how that looks on the outside as far as do we have any diseases because of that, any genetic mutations, okay? So DNA contains genes, obviously, we know that by now, right? Sequences of nucleotide bases, and I hope you guys remember those bases, because the DNA bases, we have A, adenine, C, cytosine, G, guanine, and T, thymine, okay? Those are the 4 bases for DNA. They're going to be ever so slightly different, or one is going to be different when we talk about RNA, okay? So make sure you know those base pairs, and if you don't, go back to my last lesson where I talk about those base pairs in depth. But these genes code for polypeptides. So the sequence of the nucleotide bases basically code for specific proteins, okay? Proteins are needed all over your body. When we talked about the cell membrane, remember there are proteins embedded in the membrane? Those are incredibly important because if they are misshapen or they're not made properly, then certain things can get in and out of the cell that aren't supposed to. Okay, that's a big deal. Okay, that causes for many, many, many problems and diseases and genetic variations when you have these. Well, I shouldn't say that doesn't cause for it. This genetic variation causes for those problems. Okay, so those genes code for polypeptides, which is a fancy schmancy word for proteins. Proteins are used to build cells. They do much of the work inside cells. Obviously, you have protein in the form of muscle cells. So proteins are basically all over your body, used in very different reasons, very different things, okay? So let's start off with DNA. The DNA's code must be copied, right? Yeah, that code. Remember those nitrogen bases? Those have to be coded and copied. In the cytoplasm, this code must be read so amino acids can be assembled to make polypeptides, and so those are the proteins. So again, key thing is the cytoplasm. That is where this is occurring, not in the nucleus. So once the DNA is copied, then all of a sudden you have this taking place outside in the cytoplasm, all that goopy gel, and that's where you're going to actually make the proteins. This process is called protein synthesis. Synthesis is building something up, putting something together. You're synthesizing something. We're going to synthesize proteins, okay? So let's talk about the DNA is found inside the nucleus. Proteins, however, are made in the cytoplasm of the cells by organelles called ribosomes. So we've talked about that long ago in cell structure, okay? The whole ribosomes, you know, those little studded things, they stud the endoplasmic reticulum, but also they're free-floating ribosomes. Those make proteins. Ribosomes may be free in the cytosol or attached to the surface of the ER. I'm a little ahead of myself, you know, I just kind of keep blah, 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 and then I look down at my lesson. So let's talk about the specific roles of RNA and DNA before we get into the actual protein synthesis. DNA is the master plan. Look at this. Isn't this beautiful? The master plan for the city. So DNA is the master plan for your body, for your cell, but ultimately then for your body. RNA is the blueprint of the master cell. blueprints are absolutely necessary to get to this master plan, right? You need a blueprint. So not one or the other is more important, but the blueprint is kind of like, hey, what should we follow in order to get this master plan? Okay? So remember those two concepts. RNA is the blueprint, DNA is the master plan. So RNA has some differences. So RNA is ribonucleic acid, where DNA is deoxyribonucleic acid. So if you notice, the two sugars are slightly different. RNA has the sugar ribose, and DNA has the sugar deoxyribose. So here are they, these two molecules in molecular form, okay? You have the deoxyribose, and let me guess, you probably can hardly tell the difference, right? And then the ribose right here, okay? You can maybe start to point out, oh, there's a difference, and let's see, where's the other difference? I think that's the main difference right there, yep. So the hydroxide ion right there. So very slight, but still a huge difference to set them apart, two different types of sugars. That's one difference, okay? Some other differences are, which something that you need to know for your test, RNA contains the base uracil while DNA has thymine. So what you need to understand is that RNA will not contain T. Instead, you will see U, okay? So it will be paired up with the opposite on the other side, but RNA never contains thymine. Instead, in place of, it has uracil, okay? And I showed you a video that had this little sad uracil. He was like a little teardrop, and he was crying because he couldn't play in the fun games with the DNA, because DNA does not contain uracil. So these two are swapped. So instead of thymine, RNA has uracil. The last difference, and we'll show you some base pairs so you guys can practice kind of what goes with what. So the RNA molecule also is single-stranded. Remember, DNA is a double-stranded. It's a double helix, okay? RNA, one single strand, okay? Nice and simple, okay? So those are the three differences. You guys have to know those differences between DNA and RNA. So we have different sugars, okay? Ribose versus deoxyribose. We have the uracil in RNA instead of thymine. And then we have a single strand versus double-stranded, okay? Those are the three things that you guys need to remember. Rewind it if you need to. Okay, so here's the structure of RNA. So again, the base is uracil instead of thymine. Okay, so you have to see that it's going to pair up ever so slightly different. Okay, so you have the ribose, you have one nucleotide, you have G, U, A, and C. Okay, so this is the ribose sugar. Remember, this is that kind of half of that rung, but we don't have the other half because it's only single-stranded. Okay, so we have guanine, uracil, adenine, and cytosine. Okay, so looks very similar to DNA, but again, it's single-stranded, and you have the uracil right there, and then it's only a ribose sugar instead of a deoxyribose. So there are three types of RNA. Three, yes, three. Okay, first one is messenger RNA. Messenger RNA, also called just mRNA, so you'll see that often, copies DNA's code and carries that genetic information into the ribosomes, okay? So basically all it's doing is it's taking that DNA code and it's copying it, carries it to the ribosome, because then, so messenger, like I'm gonna send this message here, and the message is the genetic information. That's all the mRNA does. The ribosomal RNA, which we call rRNA, along with protein, makes up the ribosome. So actually, that type of RNA is really what's making up the ribosome, along with proteins, okay? And then the transfer RNA. So remember, you have the copied DNA's code. It's carried to the ribosome, which is made up of ribosomal RNA. But then the transfer RNA then is going to transfer the amino acids that have been made with that messenger RNA, okay? And it's going to make those amino acids, and it's going to transfer that to the ribosomes where the proteins are synthesized, okay? So transfers amino acids to the ribosomes where the proteins can be made. So these names are very fitting. You're bringing a message to the ribosome, which is made up of ribosomal RNA and protein, and then the transfer RNA is going to bring amino acids to those ribosomes where those codes are, and it's going to code for certain proteins that can be made and synthesized. Okay? Three types. So let's say that one more time. Messenger RNA, the blueprint for construction of an RNA. So it's that messenger. What is it going to look like? What are those proteins going to look like? Okay? Ribosomal RNA is basically the construction site. Those are those ribosomes. That's where the construction is going to take place, and the protein will be made. Okay, here's a construction site. Transfer RNA, good old FedEx, right? It's the truck delivering the proper amino acids to that site at the right time. All three of these have to work together in order to have your amino acids and your proteins to be synthesized, okay? Because again, amino acids is what makes up a protein. Remember that too. Messenger RNA, so long straight chain of nucleotides made in the nucleus, okay? So it copies the DNA in the nucleus and then it comes out to the ribosomes. So that's where the protein synthesis takes place, is out in the cytoplasm on the RNA or on the ribosomes, okay? But first, you have to get that RNA to basically get that code from the DNA. Make sense? Copies the DNA, leaves through those nuclear pores. There are pores in the nucleus, in that nuclear membrane. Okay? Contains the nitrogen bases A, G, C, and U. So no thiamine. So here would be your messenger RNA. Okay, here's just a single strand, and it's going to go into the nucleus, it'll be in the nucleus, and copy that DNA code and take it out through the nuclear pore. So here's a genetic code. I want you guys to just kind of get used to seeing this. So you use the code by reading from the center to the outside, okay? So basically, remember, we have these bases. We have A, C, U, and G. Okay, there's those four bases there, okay? And if you have a code, so basically if you look for, what is the code for AUG? Okay, so here's the example. Codes for methionine. So you want to start with the first letter. So you're going to work your way. So this large letter is going to be that first letter. So A, and then your second letter is going to be U here, and then you go down to G, which would be methionine. So 3 base pairs code for 1 amino acid. So each 3 of those base pairs, you're going to have these different amino acids that then again, all of a sudden these amino acids are going to come together and be synthesized into a protein. So it's interesting that it comes down to those base pairs. You cannot have any kind of mix up with those base pairs, otherwise you get the wrong amino acid, and then you have that wrong amino acid synthesizing a protein, which is ever so slightly off. So we can try some other ones. I want you guys to look at these, okay? So name the amino acids for GGG. So again, first letter start here, G, move to this second G, and then this G is glycine. Okay, obviously all of these, if you had GGU, GGC, GGA, GGG, are all going to code for glycine. Obviously that's a very important amino acid, so that's going to have lots of different codes. How about UAC? Okay, I'll give you just a second to kind of look that over. Okay, if you follow this U, and then the C, and the A, you get serine. Okay, CAU. So again, I just want you to kind of practice this. You start from here, C, A, and then go down to U. And you have histidine. I can barely read that. Sorry, it's very small. So that is basically how you can see. Now, I mean, there are so many more codes than this. This is just showing you some of them. Well, I shouldn't say that. Those are all of the amino acids there. So actually, you do have all the codes, but it's interesting that amino acids paired with different amino acids, there's still so many different combinations when it comes to protein synthesis, okay? So those are the amino acids that are basically coded for with those 3 nucleotide bases, and then put those amino acids together and you get a protein. So remember those complementary bases we talked about. So DNA, you have adenine that goes with thymine and cytosine that goes with guanine. Well, remember on RNA you don't have T. So here's where I want you guys to remember that on RNA, adenine is always going to pair with uracil, and cytosine is always going to pair with guanine. So if you have a nucleotide base, and all you have are on the DNA, you have A, T, C, G, okay, kind of written down here, and I would write on here, but it would look like I'm a kindergartner. So if you had A, T, C, G going down, then you would have, instead of T for that A, you would have U. Okay, and then for the T, of course, you'd have A. For the C, you'd have G, and for the G, you'd have C. Okay, so I know that probably didn't make sense because you're just listening to me talk. But just know that anytime you are having a complementary base, the T on the RNA will not exist. It is always uracil, U. Okay, so, and I'm hoping if you guys have any questions on that, please ask me because you need to know that for your test. So those base pairs, remember, we learned what is for DNA. There's one change in the RNA, and that is uracil instead of thymine. So So let's talk about the transfer RNA. The transfer RNA is kind of cool. It's like this little clover leaf shape, you know, little molecule kind of thing. Yeah, that's what I call it. So basically, you have the amino acid that attaches up here. So it's a clover leaf shape, single-stranded molecule with an attachment site at one end for the amino acid. So the amino acid would be right here. Now here's the anticodon. So the anticodon has 3 bases down here that basically are going to then fit with that RNA. Now remember, you've gotten that code from the DNA, and now you have your RNA code. It's going to contain uracils in it. Well, then you have the anticodon that's going to then make it a double-stranded, and this is going to have an amino acid on top. So basically, you can have an amino acid that's attached up here. This anticodon attaches to the RNA down here, basically that messenger RNA that has carried that, that code to the rRNA. And you're going to have many different tRNAs that line up. And so you'll have amino acid and amino acid, and you'll have all these little clover leaf shaped things. So you'll have all these amino acids that start to line up and then will form a protein. Okay? So the transfer protein is kind of cool looking. It's that clover leaf shape. Make sure you guys read up in your section, okay? Because this section has some good visuals for you guys along with this. Obviously, I give you kind of a general big picture. Go to your lesson for a little bit more detail, okay? Because obviously there are some details that you're going to need to know for the test. But I like to give you guys the big picture so that you're like, okay, I get it. Now I can go to my lesson and I can understand it more and take in some more of those details, okay? Pathway to making a protein. This is a simple way to think about it. So obviously it's kind of a windy road to get to a protein, but you have DNA, and then messenger RNA is going to then copy that, right? Copy that code into the RNA code, which will have uracil in it, which then will be brought to, which is not it's not said here, but will be brought to that rRNA, so the ribosome. Then the transfer RNA will come to those ribosomes and connect to that messenger RNA with those anticodons, and then the amino acid will be attached on top, okay? And ultimately with those amino acids, you synthesize a protein. Yes, you. All right, so protein synthesis in a nutshell. The production or synthesis of polypeptide chains, which is proteins. 2 phases, transcription and translation. I'm going to let you guys look at kind of a simulation, and I have already explained transcription and translation, although I didn't specifically say those terms. You're going to understand what those mean in just a moment. So watch this.
With computer animation, we can enter the cell to view this remarkable system at work. After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a 2-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins. Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
So I really think that that's a great visual for you guys to see what's happening with transcription, which is basically taking that DNA and using the messenger RNA to get that code, take it out of the nucleus, okay, to that ribosome, and then transcription takes place where you basically are synthesizing those proteins with transfer RNA, okay? So hopefully that gave you guys a good picture. Rewind it if you need to see it again, because it shows the transfer RNA, kind of that clover leaf looking, and it's building. If you notice, it's kind of latching on with those anticodons, but then on the bottom you can see that protein being synthesized with those different amino acids, okay? So here's kind of another quick visual. Visuals are always good, right? You have your DNA, okay? The DNA unwinds with the help of this RNA polymerase, which is an enzyme. So you always need some kind of an enzyme to get something going. So that's going to unwind the DNA, and then that messenger RNA is going to come and grab that code, okay, for that particular section. It will then go out of the nucleus. If you notice, it's going to go out through this pore, and the messenger RNA will then go to the rRNA, which is kind of the ribosome, okay? So right here, we're at the ribosome. This is the rRNA. And now you have the transfer RNA that comes in, and it basically attaches to these, with these anticodons, attaches to those sites here with the amino acids attached up here. And many amino acids make a polypeptide, which makes a protein, okay? So you can see all here, here's the RNA, transfer RNA, the amino acids that bind to it, the anticodon, and they come here and they form your protein, okay? So transcription takes place basically in the nucleus, where your messenger RNA is replicating kind of what the DNA says, although with the one change, the uracil. And translation is happening where basically you are making those proteins, okay? I think I mixed those up before. So transcription is first. I think I said translation is first. Sorry if I stand corrected. Transcription is first in the nucleus. Translation is next, okay? Let's go one more thing, gene mutations. So gene rearrangements when an entire gene moves to another location, okay? You have a gene alteration when the wrong amino acid is placed during protein synthesis. You have insertion when a large part of the DNA is inserted into a gene, and you have deletion when a whole segment is lost, okay? All of these are mutations. So if something's rearranged to another location, you're going to have something go wrong, an alteration. If the wrong amino acid is there, you're obviously going to have something wrong with your protein. If you have too much DNA that's inserted somewhere, that's too much genetic information, so there's going to be a problem there. If you're missing a gene, there's going to be a problem there as well, okay? So now the last thing I'm going to show you guys, and then I will be done with section 5, is I just want you guys to listen to someone else who is dealing with a genetic mutation actually talk about it. So watch this and then we will be done.
When I found out I was BRCA positive, I did not know how to tell people.
Right.
I have a genetic mutation.
Right.
What is that?
You can start by saying everyone has genetic mutations. Mine isn't a gene we know about, right? All cancer is genetic. Some of those genetic changes, the first ones, are inherited. We get one copy of every gene from our mother, one copy of every gene from our father. Your mutation would have come through your mom. A mutation is something that has gone wrong with DNA. The genetic code is a code of words, each of 3 letters. My favorite examples is the fat cat saw the red dog run out. You take out the FA of fat. The rest of what you have in that sentence is now nonsense. Meanwhile, a person with this mutation is completely healthy. There is nothing wrong with having one copy of a mutant BRCA1 or BRCA2 because you have the other copy guarding your genome in your breast cells and your ovarian cells. All that it takes is for the remaining normal copy to be lost, for a cell to begin to become a tumor. That's why one is so predisposed to the illness, because the first mutation is already present.
So there's some good information in there in that short little video to show that, first of all, every bit of cancer is genetic, meaning not that you get every bit of cancer from, well, you do get it from your parents, you can have a gene, but it's found on your genes, most importantly. It's because there's a mutation in your genes. But if you notice, there were so many other outside environmental hazards that can cause cancer. So it's not just necessarily, oh, my dad has cancer, I'm going to get cancer. Sometimes it's how you protect yourself and you protect your DNA. Because remember, the sun's radiation, the harmful air sometimes, the stuff that we put in our bodies as far as food, all of those things can still affect our DNA. And if we have one copy that's kind of rough because you got that from your parents, you know, you got to protect that other one. Okay, so there's some good thoughts in there. So I will leave you with that. And the next time we meet, we'll be talking about our last bit of genetics. All right, have a lovely, lovely day.